वेलकम बैक टू अनदर वीडियो फ्रॉम शोमुस बायोलॉजी अनदर बायोलॉजी मैथ प्रॉब्लम्स। और इस वीडियो में हम बात करने वाले पेडिग्री मैथ प्रॉब्लम्स के बारे में। देखो पेडिग्री इज अ मैथ प्रॉब्लम जहां से सीएसआईआर नेट में हमेशा क्वेश्चन आता है। नॉट ओनली इन सीएसआईआर नेट, यू कैन गेट क्वेश्चन इन आईसीएमआर, डीबीटी जीआरएफ, आईआईटी जैम, इन ऑल दिस काइंड ऑफ एग्जामिनेशंस। And there are plenty of pedigree problems. I've already solved it many times earlier in my channel itself. But this time I'm going to solve the problem from a different perspective for the exam perspective. तो इंग्लिश हिंदी मिला के होगा, इंग्लिश में ही ये वीडियो होगा, एंड हियर आई विल एक्सप्लेन थिंग्स इन इंग्लिश एंड हिंदी मिक्स्ड लैंग्वेज. So let's start with some basic and simple category of problems, and then we'll move into a little complicated category. Basically, I need to teach you two things. One is that by looking at a pedigree, how you apply your knowledge to understand what category of the pedigree is. Generally, the category of pedigree can become either autosomal dominant, autosomal recessive, X-linked dominant, or X-linked recessive. And there are two more options: Y-linked and cytoplasmic, that is mitochondrial inheritance. But mitochondrial inheritance and Y-linked are generally not asked in the exam because they are very easy to depict and detect. तो जनरली तो क्वेश्चन आएगा वो ऑटोसोमल डोमिनेंट, ऑटोसोमल रिसेसिव, एक्स लिंक्ड डोमिनेंट, एक्स लिंक्ड रिसेसिव से होगा. सो एज अ एस्पायरेंट यू शुड बी एबल टू अंडरस्टैंड व्हाट कैटेगरी ऑफ द पेडिग्रीज, कौन सी कैटेगरी में बिलोंग करता है वो आपको ढूंढना आना चाहिए. सेकेंड इंपॉर्टेंट थिंग, यू नीड टू बी एबल टू पुट द जीनोटाइप. आपको पेडिग्री में जीनोटाइप को फिल करना आना चाहिए. एंड द थर्ड थिंग, you need to be able to calculate the probability and the risk calculation. These are the three things that you need to learn about pedigree problems. अगर इस तीन चीज़ों को पता चल गया आपको, then you will be the master of solving pedigree problems. That is what you need to do. So यहाँ पे जो examples अब लेंगे वहाँ पे तीनों ही चीज़ को हम ट्राई करेंगे एक एक एग्जाम्पल देके. So starting with this one, it stated what is the genotype of individual. यहाँ पे genotype calculation का question है, right? Find out the genotype of individual of 2, 3, मतलब second generation की third individual. So third individual of the second generation. What is the genotype of this individual? This is one question. Now, why I choose this question? Because this is a simple question. But from this question, we not only about to understand what category, what kind of pedigree it is, but also will be able to understand how to put genotypes in a pedigree. दो ही चीज सिखा देंगे एक ही एग्जाम्पल से. So first of all, we apply our knowledges of pedigree, and the biggest knowledge of pedigree I'll write it down here. The first knowledge, the first tip here, ये जो टिप है ये गोल्डन टिप है, ये चीज याद रखना. एक is that what you need to see is the parent generation and offsprings, parent generation and offspring, parent generation and offspring, and between the parent generation and the offspring's generation, if the unaffected parents give affected children, give rise to affected children, unaffected parents give rise to affected children. दोनों ही mother father both are unaffected but they give rise to an affected children. In that case, that is an example of dominant pedigree, right? No. Here I'll give you few rules. Here I'll give you few rules to incorporate. जिस रूल को incorporate करके आप जल्दी से जल्दी पेडिग्री के एनालिसिस में आंसर लेके आ सकते हो. That is the very first rule is, if this is the scenario, two scenarios I am mentioning here. Two scenarios I am mentioning. Okay. So let's say both the parents are affected, but their next generation is unaffected. Right? Both parents are affected, next generation unaffected. That is an example of dominant pedigree. And if both parents are unaffected but they have affected children, that's an example of recessive pedigree. Okay, that's the example of recessive pedigree. ये दो चीज याद रखना. Third important thing is that if a trait is transferred from father to daughter, then that is X-linked, because a father always provides X to its daughter. If the gene is transferred from, if somehow you see a gene and a son is affected, father is affected but a son is affected, then that is never an X-linked, that is always an autosomal inheritance. ये 4 चीज याद रखना. If you know these 4 things, and one last thing is that mother is affected and next sons are affected from mother, that is generally X-linked recessive. Okay, these are the 5 rules. I said this is the 5 golden rule in order to interpret any pedigree. अब इंटरप्रिटेशन हो जाएगा, आपको मतलब पता चल जाएगा कि हाँ ये ऑटोसोमल डोमिनेंट, ऑटोसोमल रिसेसिव, एक्स-लिंक्ड डोमिनेंट, एक्स-लिंक्ड रिसेसिव. बट प्रॉपरली अगर उसको करना है तो जीनोटाइपिंग आना चाहिए. विदाउट नोइंग जीनोटाइपिंग यू कैन नॉट बी 100% श्योर, बट 90% तक आपको पता चल जाएगा कि क्या होना चाहिए, और उसी एजम्पशन के ऊपर बेस करके आप जीनोटाइपिंग करेंगे. बिकॉज टू डू जीनोटाइपिंग यू नीड टू एज्यूम द पेडिग्री फॉलोइंग सर्टेन पैटर्न. विदाउट नोइंग दैट यू कैन नॉट डू इट. सो दीज आर द 5 रूल्स. एक बार हिंदी में बोल देता हूं. पेरेंट्स अफेक्टेड है मगर चिल्ड्रन अफेक्टेड नहीं है तो वो डोमिनेंट होता है. पेरेंट्स अफेक्टेड नहीं है, कोई भी पेरेंट्स, अब माँ बाप दोनों ही सही है मगर चिल्ड्रन अफेक्टेड है, दैट इज रिसेसिव कैरेक्टर. और एक केस जहां पे फादर अफेक्टेड है और फादर से सारे ही नेक्स्ट जनरेशन में, अगर ऊपर की जनरेशन में फादर अफेक्टेड है, नेक्स्ट जनरेशन में वहीं के सारे जो डॉटर्स हैं वो सब अफेक्टेड है. दैट इज एग्जाम्पल ऑफ एक्स लिंक ट्रेट, बिकॉज फादर एक्स देते हैं अपने डॉटर को और वाई देता है अपने सन को. और अगर फादर अफेक्टेड है, नेक्स्ट जनरेशन में सन भी अफेक्टेड है, तो वो कभी भी एक्स लिंक नहीं हो सकता. दैट इज रिलेटेड टू द ऑटोसोमल. और एक केस जब महिलाओं से मतलब मदर से ट्रांसफर ज्यादा सन में हो रहा है. सो इफ अ मदर इज अफेक्टेड, नेक्स्ट जनरेशन ऑल द सन्स आर अफेक्टेड, दैट इज एक्स लिंक्ड रिसेसिव ट्रेट. एक्स लिंक्ड रिसेसिव ट्रेट का यही ट्रेडमार्क है कि वो मदर टू सन ट्रांसफर करता है, और एक पॉपुलेशन में जितना अफेक्टेड इंडिविजुअल है उनमें से मैक्सिमम पुरुषों को वो अफेक्ट करता है. मेल्स आर अफेक्टेड, मेल्स विल बी अफेक्टेड. सो दीज आर द लिस्ट, दीज आर द 5 इंपॉर्टेंट टूल्स दैट यू नीड इन ऑर्डर टू आंसर द क्वेश्चन. नाउ वही रूल यहां पे अप्लाई करेंगे. सो विल यूज अप्लाइंग दिस 5 रूल्स in all the pedigree in order to find out and make an assumption what kind of trait it can be. You can see here that in this case this part is totally affected, you don't have any clue. यहाँ पे देख ले तो mother is affected, in the next generation from mother son is also affected, another daughter is also affected, right? And in this case father affected, son affected, but mother is also affected. So basically it's very difficult for us to tell कि हमारे फॉर्मूला यूज करके, जो टिप्स यूज करके, कि ये किस प्रकार के पेडिग्री है. एक चीज हम याद रख सकते हैं यहाँ पे, वन थिंग वी कैन ऑलवेज अंडरस्टैंड एंड से दैट दिस पेडिग्री इट कैन बी लाइक डोमिनेंट और रिसेसिव, ऑटोसोमल और एक्स लिंक्ड. वेरी वेरी डिफिकल्ट टू प्रेडिक्ट. यहाँ पे वेरी डिफिकल्ट हो जाता है प्रेडिक्ट करना फॉर दिस पर्टिकुलर पेडिग्री, एटलीस्ट वी से, राइट. But let's try to do it. Let's take it as a see. But one thing we can tell is that कि यहाँ पे इस generation में देखो, this is an affected individual, but the next generation there is no affected individual. But इस generation की overall बात करें तो every single generation you will find an affected. So it's not skipping that much generations, right? So let's try to put it as a autosomal dominant trait, because बहुत सारे लोग affected हैं और हमें कुछ पता नहीं चल रहा है with our general tips. तो आशा कर लेते हैं कि यहाँ पे एज्यूम कर लेते हैं कि ऑटोसोमल डोमिनेंट. अगर ऑटोसोमल डोमिनेंट ही है तो पता करते हैं कि कैपिटल ए और कैपिटल ए स्मॉल ए दोनों का मतलब ही होगा डिजीज. स्मॉल ए विल बी द ओनली ऑप्शन व्हेन देयर विल बी नो डिजीज, है ना. सो लेट्स सी इफ दैट इज द केस. सो इफ दिस इज कैपिटल ए या फिर ये कैपिटल ए स्मॉल ए हो सकता है, दिस वन इज स्मॉल ए. सो एनी इंडिविजुअल without the disease should be small a, homozygous recessive for that. So let's see here again, if that is the case then it is small a. This is, let's say again capital A small a, right? There is no other option. This is also capital A small a, right? And here also small a. If that is the case then the chances are that we get capital A small a is there, but there is no capital A small a, we are all getting small a in here. In this case also we get this or this, so we can explain any of this is possible. So with autosomal dominant it is a possibility that we can explain this this pedigree with autosomal dominant. ठीक है, autosomal dominant से हम explain कर सकते हैं. So the individual here, what is the genotype? It is small a genotype. That's we can say that it's a small a genotype. Okay, if this pedigree follows autosomal dominant category. अगर इसको autosomal recessive से देखें, now we'll try to see it via autosomal recessive category, then what will be the case? Autosomal recessive means small a is a disease and capital A small a is normal, right? So this is infected. So infected ones can be easily understood like this, okay? And the other individual should be carrier. Why it should be carrier? Because the other individual is carrier. One small a must be required in order to have another small a for these two individuals, right? So this one is capital A small a. This one is it can be capital A or capital A small a, more preferably capital A because none of these three offsprings are affected. So now if we use the autosomal recessive as a trait, then we can explain this pedigree better. If you are confusing, then please rewatch the section earlier, like when we use the autosomal dominant trait pattern to explain, there are too much ambiguity. मतलब बहुत कुछ हो सकता है, हर एक जगह पे बहुत कुछ, मतलब यहाँ पे इस टेबल पे या तो कैपिटल ए हो सकता है, या पे कैपिटल स्मॉल ए हो सकता है, जीनोटाइप का कोई ठिकाना नहीं है. बट व्हेन वी आर ट्राइंग टू एक्सप्लेन इट विथ ऑटोसोमल रिसेसिव माइंडसेट, वहां पे हमें लगेगा कि हाँ ये एक्सप्लेन हो रहा है. एंड व्हाई ऑटोसोमल रिसेसिव इज मोर प्रीडोमिनेंट? बिकॉज यहाँ पे देखो, इस जगह पे एक जनरेशन यहाँ पे अफेक्टेड है, दूसरे जनरेशन में कोई भी अफेक्टेड नहीं है यहाँ पे. ऑल्दो दिस साइड यू विल सी द सम पीपल अफेक्टेड, मगर फिर भी एक चांस लिया हमने ऑटोसोमल रिसेसिव को यूज करके. एंड व्हाट वी फाउंड आउट इज दैट ऑटोसोमल रिसेसिव becomes better option to explain this pedigree, particularly to predict the genotype of the individual 3 of the generation 2. Because if we take it as the autosomal dominant trait, तो वहां पे हो सकता है कि हेटेरोजाइगस हो सकता है होमोजाइगस डोमिनेंट हो. बट इफ वी टेक इट एज अ ऑटोसोमल रिसेसिव ट्रेट, देन वी नो दैट 100% the individual 3 of the generation 2 must have a genotype of capital S small a, which is heterozygous. So that is why autosomal recessive is more suited to explain this pedigree, and according to the autosomal recessive, the genotype of individual 3 of generation 2 must be capital S small a. Got it? So that should be your answer, heterozygous capital S small a. And now why and how exactly genotyping is done? You know, the easiest way of doing genotyping, मैं और एक बार दिखा देता हूँ यहाँ पे. The easiest way of doing genotyping is that when we know this is autosomal recessive, right? We assume that this is autosomal recessive. So when we assume that, we know small a means disease and rest anything means normal. Write it down like this. Then whoever is impacted, diseased individuals, always write their genotype because you know their genotype, right? So you can see 40% of my genotyping is done. Now we'll go backward from here, from this small a. So this one is also small a because affected, right? So there is a chance that this is unaffected, so it can be capital A small a or it can be capital A. But what it should be? If it is capital A, then all of them should be capital A small a, but that's not the case. These are small a. So to originate small a, one small a will be given by this mother, the other small a must come from this father. So this father must have a genotype of capital A small a and not capital A. So this father must be a carrier. If the father is a carrier, then only a small a will be donated to the next generation from the father. Another small a is donated from the mother, and we get a small a who are individuals affected. And this individual gets one good copy of A from the father bad copy from the mother, because mother can only give bad copy, and we'll have a genotype of capital S small a, which is heterozygous, makes this individual 3 of generation 2 as a carrier. Got it? That's how you need to think while you do the genotyping, and you need to be master at genotyping. Without knowing genotyping, you cannot solve further problems related to this kind of pedigrees. Let's move to this example now. Explain given reasons whether the following pedigrees are compatible with autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and note that the pedigree may be compatible with more than one type of inheritance as well. वो भी लिखा गया है. So again, simple pedigree analysis, you need to find out the type of trait that the pedigree is showing, okay? And it is also told that one pedigree can be explained with more than one category, more than one type of trait marking. हो सकता है कि एक ही pedigree autosomal dominant से भी explain किया जा सके, autosomal recessive भी explain किया जा सके. Okay, that's a possibility. So again, we are going to invest all those 5 tips that we learned in order to explain the pedigree category here. Okay. So I erased the data for a while, and why I am erasing the data, because I told you that there is a chance that this one is recessive, right? I told you that this is recessive trait, and I told you that this is probably an autosomal dominant trait, right? That's what we are figuring it out. Kind of the autosomal trait. It can be dominant or recessive. We'll see that later. But it is a dominant trait. It can be autosomal or X-linked. But this one is recessive trait for sure, because we have unaffected parents and affected offspring. That is recessive for sure. One more thing is that if you look at this pedigree, that only males are affected. Only males are affected. So what do you mean by only males are affected? Only males are affected in a pedigree, no females are affected. That gives us kind of an idea that it can be X-linked, recessive. For X-linked recessive trait, we know males are predominantly affected, right? But again, you can see from father, the gene is not transferred to the daughter. If the gene is transferred from the father to daughter, that is generally X-linked dominant trait. If it's a X-linked dominant trait, it must transfer from father to daughter. As here, the transfer is not from father to daughter, we can say it's not X-linked dominant, and we are sure that it is recessive, and it is also significantly saying that it is X-linked recessive. So I am going with X-linked recessive for this particular pedigree, the right hand side of the pedigree. Okay, so X-linked recessive should be our answer for this pedigree, and I am 99% sure that this is X-linked recessive based on our 5 tips that we used earlier. This is easy to answer. But for this one, we know that this can be a dominant trait, but whether it's autosomal and recessive, we need to see this out, because in the left hand side, we can see that male female are almost equally affected. Why I am calling it as a dominant trait? Because the trait comes in every single generation. So dominant trait for sure, based on its behavioral pattern, and if one parent affected, other offsprings are being affected, the parents are unaffected, the offsprings are unaffected. So kind of following the dominant category, but it can be X-linked dominant or autosomal dominant. We are not sure of that, because father was not affected. If the father was affected and all the daughters are affected, then it should be X-linked dominant, but that's not the case in here. So now we do the genotyping in order to finally understand that genotyping is super important to understand. So if we consider them to be autosomal dominant, then how we do the genotyping? That means is capital A or capital A small a, both are diseased, and small a is the healthy individuals. So any healthy individuals can be marked with small a according to the autosomal dominant pedigree pattern, right? Small a. So now if they are small a, we've written. So any individual who are healthy here will be small a. यहाँ पे देखो एक ही बात है, ऑटोसोमल डोमिनेंट हम अगर ऑटोसोमल डोमिनेंट मान के चले इस पेडिग्री को और जीनोटिपिंग करने की कोशिश करें, कैपिटल ए स्मॉल ए मतलब डिजीज होगा, और स्मॉल ए मतलब डिजीज नहीं होगा। तो कोई भी इंडिविजुअल जिसको डिजीज नहीं हुआ है वो स्मॉल है, वो हमने पहले मार्क कर दिया। नाउ सेकंड स्टेप इज टू फाइंड आउट द अदर्स। तो व्हाट अदर्स आर, सो देखो यहाँ पे ऑप्शन हो सकता है इट इज कैपिटल ए और कैपिटल ए स्मॉल ए, बट अमॉन्ग द टू विच इज करेक्ट? सी यहाँ पे वन इंडिविजुअल दिस गेट अ कैपिटल ए, so this individual, so this is okay, right? Small a, this individual was small a, normal. To get a small a, this mother must have a small a instead of having both the capital A's. तो यहाँ पे जैसे कि ये बाहर से आ रहा है एक्चुअली, यहाँ पे ये वाला है, ये वाला नॉर्मल है, ये वाला नॉर्मल है, तो कैपिटल स्मॉल ए है, दो स्मॉल ए है, एक स्मॉल ए आएगा फादर से, एक स्मॉल ए आएगा मदर से। फादर एक स्मॉल ए दे रहा है, ठीक है, मदर एक स्मॉल ए देना है, तो मदर की जीनोटाइप में एक स्मॉल ए होना ही चाहिए। सो मदर मस्ट हैव वन स्मॉल ए, राइट, इंस्टेड ऑफ हैविंग कैपिटल ए, तो एक्चुअल जीनोटाइप इसका मदर का हो गया कैपिटल ए स्मॉल ए, हो गया। यहाँ पे भी वही चीज देखो कि यहाँ पे इस इंडिविजुअल की बात करें तो यहाँ पे भी कैपिटल ए स्मॉल ही आएगा क्योंकि मदर की वही जीनोटाइप है। अभी अगले जीनोटाइप में यहाँ पे सारे के सारे कैरियर्स होंगे क्योंकि एक स्मॉल ए तो ये फादर देगा जो कि नॉर्मल स्मॉल ए, और कैपिटल ए जो है ये डिफेक्टेड मदर देगा, that is capital A small a. So we can explain this graph, we can explain this pedigree very easily with the help of autosomal dominant trait. तो autosomal dominant से explain हो जाता है, so that is autosomal dominant trait in this particular pedigree, we have explained it. And in this pedigree in the right hand side we told you it's a X-linked recessive. अब उसको करें X-linked recessive में X-linked को कैसे मार्क कर, basically autosomal pedigrees are marked, genotypes are marked based on A B, कैपिटल ए मतलब डोमिनेंट, स्मॉल ए मतलब रिसेसिव, उस हिसाब से करते हैं। फॉर अ एक्स लिंक कैरेक्टर वी मार्क इट लाइक दिस, एक्स ए, राइट, एक्स ए एक्स लाइक दिस, एक्स ए वाई लाइक दिस, इस हिसाब से हमें मार्क करना होता है। सो इफ दिस इज एक्स लिंक रिसेसिव दैट मीन्स एक्स ए वाई मीन्स डिजीज, क्योंकि मेल्स गेट ओनली वन एक्स, so if they get the defective gene they'll have a disease, and in females they'll only get disease if there is XA, then only there will be disease. Apart from that any other genotype won't consider any disease. So individuals with the disease is XAY, let's say this one, and XAY this one, and this one will be XX, and this one is also XX. XX नहीं XAX होगा ये, क्योंकि one XA is donated by this father but the other X is donated by the mother, right? So it has XAX, right? But as this is a recessive trait, so in order to get a disease you should have XAXA, because although there is a faulty X to this female but there is a healthy copy of X that is helping the female to live normal life, okay? So that is done. And in this side also what we have, what else we have this XAY, so somebody is donating this X, so this is X and Y, this is XAX, obviously then only we will get this. And if this is the XAX, so this is XY and this is XAX, okay? So we can clearly explain the pedigree, this right hand side pedigree with X-linked recessive trait, so that is X-linked recessive formula. And now I am 100% sure that this pedigree following X-linked recessive pattern, okay? So the difference between 99 and 100% is that 99% I reached based on my 5 tips and I reached really fast. But for genotyping it takes some time, but genotyping gives you 100% confidence in answering a question. This side also I reached the conclusion of dominant type of pedigree within seconds, but then whether it's X-linked dominant or autosomal we need to assume separately and do the math, and I did the math for the autosomal dominant category, और autosomal dominant category completely explains this pedigree. Autosomal dominant pattern को use करके completely हम इसको explain कर पा रहे हैं, तो we can easily say this is autosomal dominant and this side is X-linked recessive. Clear? You can take the screenshot, and obviously I know, I understand this might feel a little difficult at first, but the more you practice it's getting better and better with time, it will get better and better in time. So do not stop practicing, keep practicing, practice करना है. अब लास्ट और एक क्वेश्चन करके कंप्लीट करेंगे इस पेडिग्री प्रॉब्लम सीरीज, एंड द प्रॉब्लम इज रिस्क कैलकुलेशन प्रॉब्लम. नाउ टिल दिस पॉइंट आई बिलीव यू अंडरस्टूड व्हाट इज पेडिग्री, पेडिग्री को कैसे सॉल्व करना है विद द हेल्प ऑफ टिप्स एंड ऑल्सो हाउ टू डू द जीनोटिपिंग इन द पेडिग्री. वो जब आपको समझ आ जाएगा देन यू नीड टू अप्लाई दैट नॉलेज इन ऑर्डर टू फाइंड आउट द प्रोबेबिलिटी और रिस्क कैलकुलेशन. Now here in this question, they ask you a question regarding the offspring out there, whether it can be male or it can be female or it can be male. Between this male or female, whatever individual came here, what is the chance of this individual to be affected by the same disease that is present in this pedigree? ठीक है, यही रिस्क कैलकुलेशन होता है. रिस्क कैलकुलेशन प्रॉब्लम में ये पूछा जाता है कि ये प्रकार की एक बीमारी है और ये बीमारी होने की चांसेस और संभावनाएं इस XYZ इस इंडिविजुअल को कितना है. दैट इज द रिस्क कैलकुलेशन प्रॉब्लम. सो यू नीड टू अप्लाई द नॉलेज ऑफ प्रोबेबिलिटी रूल्स इन अ पेडिग्री एनालिसिस. सो दोनों चीज को एक साथ कंबाइन किया जाता है. फर्स्ट इज the solving of pedigree and second is the probability rule. Then only you'll get to the answer in here. Okay, so we need to figure it out what kind of problem it is. We need to figure it out with the help of this risk calculation. And first thing that you need to find out is the trait, what kind of trait it is, then you'll find out the rest. So autosomal or X-linked, we don't know, but we know one thing, that the parents are unaffected and they have affected offspring. So if parents are affected and the offspring is unaffected, that is a recessive trait. That's a mark of a recessive trait. So this is a recessive trait, that we know that for sure. Now this recessive trait can be either X-linked recessive or autosomal recessive. We need to assume that. Sometimes in the question paper they will tell you that this is autosomal recessive or X-linked recessive in order to make it simpler for you. But here they didn't mention. But one information they must mention in here, I am mentioning it. So any individual coming from outer lineage or outer pedigree are dominant in nature. Dominant genotype means homozygous dominant genotype in nature. That means apart from this particular family's lineage, this individual and this individual, they came from outside, they are outsiders. So they have a dominant genotype. This information is very important, otherwise there will be too much ambiguity in order to answer the question here in this case. Okay. So once you know that, and we know that the parents are affected, the children, parents are unaffected, children is affected. So it's a recessive trait. Let's say assume there's an autosomal recessive trait. तो ऑटोसोमल रिसेसिव ट्रेट को पकड़ के ही इसकी सोल्यूशन हम निकालेंगे. और यहाँ पे देखो, अगर ऑटोसोमल रिसेसिव होता है तो स्मॉल ए इज द डिजीज, कैपिटल ए और कैपिटल ए मींस नो डिजीज. ओके, सो स्मॉल ए इज डिजीज, लिख दिया. एंड वी नो दैट दिस पेरेंट्स आर heterozygous because they are carriers, so the parents are carriers. And it is also told that any individual involved in marriage to this particular family from outside have carrying a dominant genotype. So they all carry this dominant genotype, capital A. These two. Generation 2 ki 3 and 4, these two individual have a capital A. This is already mentioned. So if that is mentioned, what we get from here is that what is the chance of this individual? The chance either it can be capital A or it can be capital A small a. ठीक है? So if it is capital A, there is no chance that this individual is a carrier, right? This individual is possible for this individual to be a carrier is only when this is capital A small a. इसको capital A small a होना है, तभी जाके इस इंडिविजुअल को कैपिटल ए स्मॉल ए होने का मौका मिलेगा. बिकॉज दिस मदर विल गिव अ कैपिटल ए, फादर मस्ट गिव अ स्मॉल ए, सो फादर मस्ट बी अ हेटेरोजाइगस इंडिविजुअल और अ कैरियर, राइट? सो व्हाट इज द प्रोबेबिलिटी दैट दिस फादर इज अ कैरियर? क्या प्रोबेबिलिटी है बिटवीन कैपिटल ए और कैपिटल ए स्मॉल ए? दो ही रीजन था, तो 1 out of 2 टू बी अ कैरियर. एंड व्हाट इज द प्रोबेबिलिटी ऑफ दिस बिकमिंग अ कैरियर? Half, also half, right? If this father is a carrier and the mother is capital A, what is the probability that this individual is a carrier? Try to understand. Father here is capital A small a, mother here is capital A. इनमें से क्या क्या ऑप्शन आएगा? कैपिटल A small a, कैपिटल A small a, कैपिटल A. So what is the probability of this heterozygous? Again 2 out of 4, that is half. That's what I wrote. So the probability of this individual being a carrier is half. Probability of this individual 1. So individual 2 of 2 to be a carrier is half. Individual 1 of 3 to be a carrier is also half. तो total यहाँ पे probability हो गया half into हाफ 4 इस साइड में. And in this side, again the same situation will run, right? This is heterozygous. The chance of being heterozygous here is half. This chance of being heterozygous is half. So that is half multiplied by half. So in the generation number 3, now we come to the last point here. In the generation number 3, the probability of 3A, C1 to be a carrier is 1/4. And the probability of this 3(2) is also be a carrier is also 1/4. और अगर ये दो कैरियर ना बने तो यहाँ पे नेक्स्ट जनरेशन कोई इन्फेक्ट नहीं होगा. फॉर द नेक्स्ट जनरेशन, दैट विल बी द जनरेशन नंबर 4, फॉर एनी मेल और फीमेल टू बी इन्फेक्टेड, both the parents in generation 3 must be a carrier. The chance of becoming a carrier for both these parents and generation 3 is 1/4 and 1/4. And if they are carrier, then, so this is the genotype. So what is the chance of being infected? बता, infected मतलब disease मतलब small a, that is 1 out of 4. If they are carrier, then the chance that the offspring will be infected is 1/4. So when we count the overall probability of this offspring to be infected, their offspring to be infected 1/4 when their parents are carriers or heterozygous. What is the probability that their parents are heterozygous? 1/4 into 1/4. We will do the multiplication. Multiplication rule will be applied for risk calculation problems. हमने पहले भी बात कर चुके हैं, मल्टीप्लिकेशन रूल यूज करके हमने रिस्क कैलकुलेशन का बेसिक प्रॉब्लम सॉल्व किया जेनेटिक्स में. सो दैट विल बी टोटल ऑफ 64. 1/64. 1/64 should be the correct answer in this particular question. That's how you can solve it. To count any probability or risk calculation, to count any probability of a disease in a population, you need to count the probability of the parents in order to provide that genotype to the offspring. क्योंकि वो parent के ऊपर depend करता है. यहाँ पे ये जो इंडिविजुअल है इन्फेक्टेड होगा कब? होमोजाइगस रिसेसिव. होमोजाइगस रिसेसिव क्यों होगा? अगर पेरेंट्स ये 1 और 2, ये दो थर्ड जनरेशन की अगर हेटेरोजाइगस है तो ये हेटेरोजाइगस होने की चांस हमने कैलकुलेट किया 1/4 आया. और तब जाके if those two individuals are heterozygous, then there is a chance कि offspring homozygous recessive होगा, उसका चांस होगा 1/4. So 1/4 into 1/4 into 1/4 gives us 1/64. That is the net probability for this individual mated by the third generation individual 1 and 2 to be infected. That is, it sounds little complicated. I believe it's fine. Rewatch एक बार नहीं तो 2, 10 बार, 15 बार, 20 बार, until unless it gets clear. It will get clear. और क्या करना है ना, इस टेबल को जैसे मैंने पहले पिक्चर दिखा उसको यू नीड टू टेक अ स्क्रीनशॉट ऑफ इट और ट्राई करना है, खुद से ट्राई करना है. जब देखोगे तो आसान लगेगा, इट्स डन, इट्स हो गया. बट करने जाओगे तो प्रॉब्लम होगा. वहीं पे आपको कहाँ पे अटक रहे हो वहीं पे चेक करना है. रिकॉल इज मोर इंपॉर्टेंट. So if you want this kind of problems, more and more these problems, हजारों ऐसे problems है. The book in Biomath handbook हम सेल करते हैं. The one of a kind book from Shomu's Biology only. You need to get the whole package of study material in order to get it. 3,900 प्राइस में आता है टोटल बुक सेट की पैकेज में. Or else you can join our online coaching. You will get this kind of problems explanation in the classes as well. जहाँ पे हमने dedicated class रखा हुआ है जहाँ पे ये discussion होता है. Okay, so rest is your choice. So genetics is a chapter where 80% of the question will be this mathematical or analytical, right? So for genetics, you need to have this book, Biomath Handbook. And actually, if you look at this whole book, it's approximately 250 to 60 pages book. And with this near about 260 pages book, most of the book is consisting of biochemistry math problems and genetics math problems and cladogram problems, because these are the three problems area. These are the areas from where CSIR NET ask a lot of math problems. So if you get inside, the genetics chapter starts at 67th page and ends in 128th page in edition 2. You might get edition 3 books In that case the page number might vary a little bit, but we increase the number of question in every single edition. So genetics की problem की बात करें तो यहाँ से genetics book 67, page number 67. Here we have the genetics math problems, and from here you will get Mendelian, non-Mendelian problems, genetics gene mapping problems, pedigree analysis, probability, risk calculation, additional problems, HFR mapping, bacterial genetics problem, co-transduction frequency problems. So you'll get all these problems. Here are all the normal Mendelian genetics and inheritance problems आप देख सकते हो, right? And if you want to practice more from the, let's say, if you want, so these are all non-Mendelian inheritance problems. Answer with explanations are also provided. It's not just answer, but explanations are also provided. And we have practice problems also. You will get the gene mapping problem. These are the gene mapping problems starting at page number 94 here. So these are all gene mapping problem. You can see that sample problem with answers are provided. These are the gene mapping problems. You can see in this side gene mapping problems are going on, right? Gene mapping problems. So all these gene mapping problems are there, and not only the gene mapping problems are there, but also their answer explanations. It's not likely there are 1 or 2 questions. There are more than 5 questions are there from every single topic that we discuss here, right? आप पेज scroll करते करते थक जाओगे, इतना problems आपको मिलेगा. This is another gene problem, gene mapping. It's continuous gene mapping. Now started with pedigree. यहाँ से pedigree start होता है. Pedigree problems. You can see pedigree. All these problems, pedigree problems, never ending pedigree problems. बहुत सारे problems आपको मिलेंगे, and you will get to answer them. You will get to know these problems. These are bacterial gene mapping problem, HFR mapping problem. These are also there, the HFR problems, right? So all these problems you will get from this book, that is Biomath Handbook. Okay, and there is a QR in every single chapter. If you scan them, it's going to open up the videos explaining this book. So this is a one of a kind book which you can get from us and us only, but it's not to be sold alone. You need to get the complete study package, that is of 3,900, including the delivery charges to your place.